Hello everyone, and welcome back to our training program, which is about the designing primer. So today we are going to talk about how to design primers to amplify the entire gene during a routine PCR. So here we will take the actin protein as an example of how to design a primer for a whole genome. And we will explain how to get the forward and the reverse primer for a given DNA sequence, like here the actin. And we can tell this is a gene for the actin, or in general we can tell this is a gene because it has the T for, so it refers to the DNA. And also the first three nucleotide, it's ATG. If we imagine it's the mRNA, it will be AUG. So this is the starting codon. And if we look down here at the end, the TGA, if we imagine the mRNA, that will be UGA, which is a stop codon. And this is the entire gene sequence for the actin. So how we get the primer from this? So basically there is two websites here for primer design. If we click on the first one, this will show up, which called the Primer Blast. So basically the Primer Blast is actually a website. We can use it to optimize the design of our primer. So what we're gonna do is we take the whole sequence that I already shown the previous slide, copy it and paste it in here, and then we can choose the product size for the PCR. We can, like in here in this example, we use the minimum 70 nucleotide and the maximum 1000, and we can choose the number of primers. Like here we chose 10 pairs of primers. And in here in the primer melting temperature, we can optimize the minimum TM temperature. We set it here to 57, and the maximum TM we set it for 63. And we also set the optimum melting temperature for 60, and here it can show the maximum TM difference between the minimum and the maximum. We chose here only 3. And if we want to have so many primers, we can lower that like to 50 and maximum that to 70. And we can even increase the number here for the difference to 5. And below here you will find a lot of things that you can change, but I would prefer to not change anything and leave it. Basically this procedure, it's not good if we want to amplify the entire gene, and we will show why. This is like just showing, as I mentioned before, if we want to have a lot of primers, we can change the minimum and the maximum, and this is only show how to after I change the setting. And as I said, the below sections we just don't play with it. So basically this slide showing the results. If we click on the results, we will have this chart showing up, and basically we chose 10, so I just take a picture for the first 5, but the result will be 10 pairs of primers, and I put signs here to show that this is the most optimal pair of primers, and I want to show that this is the forward primer and this is the reverse, and this is the product length is 98, and the plus it means, as I mentioned, here's the coding strand, while the minus strand is the templates, represent the templates, and the length of the primer is just a 20 nucleotide for the forward primer, and the reverse primer is also a 20 nucleotide. But as I mentioned, this is bad. Because if we go back to our genome and we find our primer, just Control and F, we will see that our primers that we designed is located in the middle. That means the amplification will happen to the right. That means the amplification will happen from here to the end, while the beginning of the gene will be lost. These sequences will be lost. And this is like showing all the sequence that will be lost if we use the primer that we designed, or if we use the first set of primer. Even if we use the second or the third or the 10th pair of primers, it will show up in the middles, will not select the whole genome. So to avoid this problem, we can use or design another set of primers using the other website that I highlighted in here. This is the page of this the second website for designing primers, and we're going to explain how to use it. Here we are going back to our genome sequence for the actin. So what we're gonna do is start from the first nucleotide in here, and we count 20 nucleotide, okay, and we copy it, and I will, as I show here, this is I changed the color for the nucleotide, and I copy it and paste it in here, and I name it the forward primer from 5' to 3'. Okay. For the reverse, we're going to the end and count backward 20 nucleotide, but we have to do extra step to get the reverse primer. So we use the other website. We click on it. First, we're going to copy the last 20 nucleotide, and then we click on the second website. It will show up this page, and then we paste the last 20 nucleotide, we paste it in here, and then we click on reverse complement. Okay, so here's our sequence. We copy that sequence, and then I will paste it in here. Then we will have the forward and the reverse primer. Okay. And if we want to make sure that we get it correct, so we compare the last 20 nucleotide with the reverse primer that we get from the second website. So we can see that the first nucleotide is T, and it's the reverse of it is A, and so on. The second nucleotide in here, A, so the opposite of it is the T. So that means we get it correct. So now we can send these two primers. We order these primers to certain companies that can make it and then we can use it later for cloning and whatever purpose we want use it. And in this slide we're showing how to get so what we Google to get this website. Basically we Google the get reverse complement, and we choose the reverse and or complement DNA sequence. After we click it, we will have this website showing up. For the other website, we're going to use this website, www.ncbi.nlm.nih.gov, and we can here type whatever gene we want to design a primer for it, and we choose here the nucleotide. Hopefully that's beneficial for you. Thank you.